Good morning, everybody. Welcome back to Daily Lectures from a New Age Cosmetic White EdTech Healthcare Consultancy. I'm Dr. Bobby. We're on the subject of molecular biotechnology, and we're specifically on the topic molecular diagnostics. We are on the subtopic of human monoclonal antibodies. Although most of the immunotherapeutic agents that have been developed have been effective, there are drawbacks to the use of monoclonal antibodies that contain non-human sequences. For example, if multiple treatments are required, which is often the case, it is desirable that the antibody contain no or only a very limited amount of non-human sequences to prevent immunological cross-reactivity and sensitization of the patient. Unfortunately, it is very difficult to create human monoclonal antibodies for a number of reasons. The human chromosomes of fused lymphocyte mouse myeloma cells during hybridoma formation are unstable, and cells that produce a human monoclonal antibody are rarely formed. To date, no human myeloma cell line has been discovered that can replace the mouse myeloma cell line in this procedure. Even if it were possible to form human hybridoma cell lines, it is contrary to accepted norms of medical research to inject humans with a specific antigen for non-therapeutic purposes. And to perform a partial splenectomy to collect antibodies producing cells. Therefore, it has been necessary to devise other approaches obtaining human monoclonal antibodies. To address this need, researchers constructed a xenomouse, which I think was mentioned yesterday. I'll just repeat it for continuity purposes, in which the one mouse antibody production machinery was inactivated and all of the human immunoglobulin loci, both light and heavy chains, are integrated into a mouse chromosome. The human heavy chain genes and human K and lambda light chain genes, where K and lambda are different classes of light chain genes, were cloned onto yeast artificial chromosomes that can carry very large amounts of foreign DNA. The YSCs with the human immunoglobulin genes were then introduced into mouse embryonic stem cells by fusing YSC-containing yeast spheroblasts with the embryonic stem cells. This procedure yields a large number of embryonic stem cells in which all of the introduced human immunoglobulin genes have become stably integrated into the chromosomal DNA. These transfected cells are used to generate mice containing human immunoglobulin gene loci. Crossbreeding of two mouse lines, one carrying both mouse and human immunoglobulin genes, and the other carrying the defected mouse immunoglobulin genes, produced a mouse strain called xenomouse. That expresses only human immunoglobulins. It is now possible, after humanization of a xenomouse with a particular antigen, to produce a fully human immunoglobulin, the large human antibody repertoire. The xenomouse has enabled researchers to produce a number of fully human antibodies, many of which are currently at various stages of clinical development. For example, the first fully human monoclonal antibody produced using this technology is Ipilimumab, which was approved in September 2006 for the treatment of certain forms of colorectal cancer. Antibody fragments. Naturally occurring antibodies are highly specific targeting reagents that provide animals with a powerful means of defending themselves against a wide range of pathogenic organisms. and toxins. Right? Immunoglobulin G, IgG, is the main antibody found in mammalian serum, and it is the native form that is almost exclusively used in therapeutic antibodies. The fact that IgG molecules have two identical sites that binds to identical antigens, that is, they are bivalent, generally increases the effectiveness in vivo, while the Fc portion of the IgG molecule is important in recruiting cytotoxic effector functions through complement or interaction with specific receptors. Fc-mediated effects are not necessarily for all applications and may even sometimes be undesirable. Over the past several years, by manipulating portions of the IgG, light and heavy chain cDNAs, researchers have constructed a variety of IgG derivatives or fragments that may be used instead of whole antigen body molecules. Some of these molecules, because of the small size, bind more efficiently to targets that are inaccessible to conventional whole antibodies. Others have multiple sites for binding to the same antigen, while others have binding specificities for two or more target antigens. Initially based on Fab and Fv fragments of antibodies, antigen-binding single protein chains, SCFE, consisting of only VL and VH domains, were developed. Single-chain antibodies may be used for a variety of therapeutic and diagnostic applications in which Fc effector functions are not required, or when small size is an advantage. Single-chain antibodies have a molecular mass of approximately 27 kilodalton compared with approximately 150 kilodalton for Ig molecules. Because of the small size, single-chain antibodies can penetrate and distribute in large tumors more readily than intact antibodies. In addition, a protein coding sequence can be linked to a single-chain antibody sequence to create a dual-function molecule that can both bind to a specific target and deliver a toxin or some other specific activity to a cell. Excuse me. Computer simulations of the three-dimensional structure of potential single-chain antibody showed that V1 and V6 domains, the VL and VH domains, have to be separated by a linker peptide to assume the correct conformation for antigen binding. On the basis of this design contract, the DNA constructs of DNA constructs of VL and VH sequences from a cDNA of a cloned monoclonal antibody were each ligated to a chemically synthesized DNA linker fragment in the order V1, VL linker, VH. After expression of E. coli, in E. coli, the single-chain protein was purified and then and both its affinity and specificity were found to be equivalent to those of the original intact monoclonal antibody. Moreover, instead of linking the VH and VL chains with a short peptide, amino acids in the FR can be modified to form a disulfide. Linkage between the two peptides, the effectiveness of this disulfide stabilized Fe molecule, VL S2S VH coupled to a cancer cell toxin was compared with that of an SCFE molecule coupled to the same toxin. The disulfide stabilized and SCFE immunotoxins have the same activity and specificity. However, the former molecule was several-fold more stable than the latter. This suggests that disulfide stabilized FE molecules may be more useful than SCFE molecules in some therapeutic applications. The two types of molecules have been used in different ways, for example, by altering the number of amino acids in the linker. In an SCFE molecule, usually to 5 or fewer amino acids, it is possible to direct the self-assembly of these molecules into either bivalent dimers called antibodies. Trimers, tribodies, or tetramers, tetrabodies, shortening the linker effects not only the multimerization but also the stability of the molecule with molecules with a shorter linker. Let's just see this continuation. After that, we just come back to the milestone note. Turning to be more stable. In addition, it is possible to combine two different antigen specificities into a single bispecific antibody. Okay, going to the milestone note. Synthesis in E. coli of a polypeptide with human leukocyte interferon activity. Courtesy is Nagata, H. Tyra, A. Hall, L. Johnsrud, Struley, M. Struley, J. Exodi, W. Ball, K. Kenter, and C. Wiesman. Courtesy and thanks to them for this invaluable milestone note. The late 70s and early 1980s were a time of tremendous excitement in molecular biotechnology. The promise of this new technology was being touted to both the public and large institutional investors. One of the products of the new biotechnology that captured the imagination of a large number of people was IFN, which at the time was seen by many as a possible miracle cure for a wide range of diseases caused by viruses and cancer. Thus, the isolation of a human IFN cDNA and its subsequent expression in E. coli were reported in newspapers and magazines around the world. Several features of IFN made it particularly difficult to synthesize and isolate a cDNA encoding the polypeptide. First, although IFN had been purified more than 80,000-fold, although minuscule amounts were available, so researchers did not even have an accurate estimate of its molecular mass. Second, unlike many other proteins, IFN did not have a chemical or biological activity that was easy to monitor. At the time, its activity was measured by the reduction in the cytopathic effect of an animal virus on cells in culture, in which was an extremely complex and time-consuming process. Third, unlike insulin, researchers had no idea if there was one particular human cell that produced high levels of IFN and therefore could serve as a source of mRNA that was enriched for IFN mRNA. These problems notwithstanding, a cDNA encoding IFN was eventually isolated and characterized. Since that time, researchers have discovered that there are several different types of IFNs. Unfortunately, IFN is not the panacea that was dreamed of by both investors and the press. However, the genes for several IFNs have been isolated, and clinical trials have shown that they are effective treatments for a variety of viral diseases. Now, most very common antibody toxin combinations, immunotoxins, have been constructed using Pseudomonas exotoxin A, which is a 66 kilodalton protein with 3 separate domains. Domain 1 is responsible for cell binding, domain 2 for translocation of the protein into the cell, and domain 3 for ADP ribosylation. Other protein toxins that have been used include bacterially derived diphtheria toxin and the plant-derived toxin rice. An immunotoxin is generally synthesized by replacing the N-terminal domain of the toxin, example Pseudomonas exotoxin A, domain 1, with a single-chain antibody sequence, thereby creating molecules very similar in size to the original toxin with the ability to bind, enter, and kill a specific cell. A number of immunotoxins that have antitumor activity in vitro and in animal models have been constructed. These include antibodies directed against the p55 subunit of the interleukin 2 receptor, the transferrin receptor, carbohydrate antigens, the epidermal growth factor receptor, and some cancer cell surface proteins. Toxin molecules may also be directed to cancer cells by using a dye-specific antibody that is engineered to bind to a surface-specific tumor-associated antigen and then to a toxin molecule, thereby directing the toxin molecule to the tumor. A number of different engineered immunotoxins are currently in clinical trials. It may be possible to create peptides that are smaller than scFvs and still retain the ability to bind to a specific antigen. The rationale for developing small antibody toxin complexes is that they are more likely to penetrate a tumor, more completely stop tumor growth. Recently, a group of researchers constructed a short peptide, 28 amino acids long, that retain the binding specificity of the monoclonal antibody from which it was derived. It was well established that antibody binding specificity resides within the 6 hypervariable loops called CDRs, 3 from the variable region of the heavy chain and 3 from the variable region of the light chain. In all antibody molecules, these CDRs are flanked by FRs. Moreover, it was speculated that, at least for some antibodies, that the major portion of the antigen binding site might reside primarily within 2 CDRs, one from the heavy chain and the other from the light chain. To test this possibility, starting with the genes for the variable portion of a monoclonal antibody against the surface antigen from Epstein-Barr virus, 8 different peptide combinations were synthesized. Each peptide contained at least one CDR3 loop, known to be the major antigen contacting segment, as well as one other CDR loop and a linker peptide, usually NFR spacer. All 8 of these peptides were tested in vitro for the ability to compete with the parental antibody for binding to the Epstein-Barr virus, thought to be the causative agent of Burkitt lymphoma and other cancers. Surface protein. One of the peptide combinations, VHCDR1, VHFR2, B1CDR3, appeared to be promising. Next, this peptide was coupled to a toxin molecule, collagen 1A, and the combination was tested both with cells in culture and with mice. In mice, the peptide collagen duct efficiently traveled through the circulation and then found and killed the tumor cells expressing the target antigen. Collagen 1A by itself does not affect these tumors to a significant extent, although the original monoclonal antibody is unable to penetrate into the tumor. On the other hand, the peptide collagen duct accumulated at the cones of the target tumors. The very exciting work is at an early stage of development, so a large number of issues remain to be addressed before it can become an effective human therapeutic measure. Nevertheless, the demonstration that a small peptide can mimic the binding specificity of an antibody molecule and successfully deliver a cellular toxin to targeted cells may provide the basis for a whole new approach for treating tumors. Combinatorial libraries of antibody fragments. Hybridoma cells, like most other animal cells in culture, grow relatively slow, do not attain high cell densities, and require complex and expensive growth media. The cost of monoclonal antibody production is an impediment to their widespread use as therapeutic agents. To circumvent this problem, attempts have been made to genetically engineer bacteria, plants, and animals to act as bioreactors for the production of monoclonal antibodies. For effective delivery and function of some immunotherapeutic agents, only the antigen binding region of an antibody, the Fab or Fv segment, is required. In other words, the Fc portion of an antibody is dispensable for some applications. An elaborate series of manipulations makes it possible to select as well as produce functional antibodies in E. coli. cDNA is synthesized from mRNA isolated from mouse antibody-producing cells, B lymphocytes. The H and L chain sequences in the cDNA preparation are amplified separately by PCR. Each amplified cDNA preparation is treated with a specific set of restriction endonucleases and cloned into a bacteriophage lambda vector. The cDNA sequences of the H and L chains each have distinct restriction endonuclease recognition sites, an arrangement that facilitates the directional cloning of each sequence into a separate bacteriophage lambda vector. At this stage of the process, many different H and L chain sequences are cloned. The cDNAs of one H and one L chain are cloned into a single combinatorial vector, thereby enabling the bacteriophage to co-express both chains, thus forming an assembled antibody Fe fragment. H and L chains are expressed during the lytic cycle of bacteriophage lambda, so that the library of combinatorial bacteriophage clones can be screened for the presence of antigen binding activity. The step in which L and H chain cDNAs are combined on one vector creates a vast array of diverse antibody genes, some of which encode unique target binding sites whose isolation would not have been possible by standard hybridomal procedures. The mammalian antibody repertoire has the potential to produce approximately 10⁶ to 10⁸ different antibodies. A phage library contains approximately this number of clones, so one combination one combinatorial library can be expected to produce as many different antibodies, in this case Fe molecules, as any mammal. In addition, once an initial combinatorial library has been constructed, it is possible to shuffle the L and H chains to obtain Fe molecules that recognize unusual epitopes, and even greater variation may be achieved by random mutagenesis. Of the DNAs in the combinatorial library, as in Chapter 8, because millions of bacteriophage blocks can be screened in a relatively short period, the identification of Fe molecules with the desired specificity takes only about 7 to 14 days. By contrast, screening hybridoma cell lines is a slow, time-consuming process. Because they lyse bacterial host cells, as bacteriophage lambda vectors are not particularly useful for production of large quantities of proteins. To overcome this drawback, the bacteriophage lambda vector was engineered so that the H and L chain DNA sequences were inserted into a site that was flanked by plasmid DNA sequences. This plasmid DNA containing an H and L chain DNA combination can be excised from the bacteriophage lambda vector and transformed into E. coli. As part of a plasmid, large numbers of Fe fragments can be produced in E. coli cells. As an alternative to the use of bacteriophage lambda, filamentous bacteriophages such as AIIMS&NEET and FD have been used for the production of combinatorial libraries. In these cases, the antibody fragment is synthesized as part of a fusion protein that is located on the outer surface of the bacteriophage. A combinatorial library of antibody fragments displayed on the surface of a filamentous bacteriophage can be screened by an enzyme-linked immunosorbent assay-like system. Briefly, samples aliquots of the library are added to the wells of a multi-well plate that are coated with the target antigen. The wells are rinsed thoroughly to remove any unbound bacteriophage. Next, an antibody that binds to the bacteriophage caused coat protein and is conjugated with an enzyme is added to each well. The wells are rinsed to remove any unbound antibody enzyme complex. The phage particles bound to the target antigen are recognized by the antibody enzyme complex. A chromogenic substrate that is cleaved by the bound enzyme is then used to determine which wells contain a phage-carrying antibody to the target antigen. This approach is easier than using block assays with bacteriophage lambda to select and subsequently purify a bacteriophage producing an antibody fragment that binds to a specific antigen. Once the desired antibody fragment producing bacteriophage has been isolated using either bacteriophage, lambda, or M13, the DNA can be isolated and subcloned into an expression vector. These procedures are used to produce mouse, chimeric, or humanized antibodies. A combinatorial library of full-length antibodies is the next topic. Until recently, all of the combinatorial libraries of antibodies included either single-chain antibodies or Fab fragments and not full-length antibodies. However, for many applications, it is advantageous for therapeutics antibodies to be full-length. With this in mind, researchers cloned and expressed complete antibody molecules using either two separate vectors, one encoding a light chain and the other encoding a heavy chain, or a single operon controlling the expression of both the light and heavy chain on a single vector in E. coli. The process of selecting antibodies with specified affinities would be greatly facilitated if an E. coli library could be screened directly for binding to various antigens. To do this, a combinatorial library of full-length antibodies was generated and engineered so that the antibodies were secreted into the periplasm between the inner and outer membranes. In addition, prior to the expression of the antibody molecules, a host E. coli was engineered to express a fusion protein that became anchored within the inner membrane lipoprotein fragment and also contained a portion of a protein from the Staphylococcus aureus protein A that binds specifically to Fc regions. When an IgG molecule is secreted into the periplasm, the fusion protein binds to the Fc region. The tightly bound Ig fusion protein complex remains intact when the E. coli cells are treated with EDTA and lysozyme to remove a portion of the outer membrane. Then a fluorescently labeled target antigen is added to detect those cells that have expressed an antibody directed against the target antigen by cell sorting. Fluorescently labeled cells are selected, then the plasmid DNA that encodes the selected IgG molecules is isolated and expressed in an E. coli strain that does not synthesize the membrane-anchored fusion protein. This simple yet powerful technique simplifies selection and production in E. coli of full-length monoclonal antibodies. Shuffling CDR sequences. Very large libraries containing of a single chain antibodies can be the sources of a wide range of highly specific human antibodies so that one does not have to resort to using mice for the initial monoclonal antibody. Theoretically, such libraries contain a greater diversity of antibodies than is normally found in the human immune system. To construct a library of this sort, B cells and other antibody-producing cells from different non-immunized individuals and from different tissues and organs are collected and pooled before the mRNA is isolated. The isolated mRNA is used to program the synthesis of cDNA, which then becomes a template for the specific PC amplification of each CDR separately. The amplified CDRs are mixed with oligonucleotides encoding FRs, a linker, and a DNA sequence encoding the variable L and H domains. Overlap extension, PCR, is used to order, join, and amplify H and L antibody chain genes. In addition to being entirely human, the 2 by 10 raised to 9 different single-chain antibodies that have been produced in this way have a wide variety of specificities, reflecting the fact that CDR sequences have been incorporated in random order and from a variety of sources, that they have been shuffled There are single-chain antibodies against the beta-galactosidase, the beta subunit of cholera toxin, fluorescein isothiocyanate, human cell surface antigen, human leptin, human prostate specific antigen. I think that's prostate. And streptavidin, among others. Moreover, the dissociation constants, K2S, KDS, for the interaction of the selected antibodies with their target antigens ranged from 0.9 into 10 raised to minus 9 to 420 into 10 raised to minus 9 m raised to minus 1, where tight binding is represented by a low number. For antibodies, K2S of around 0.1 and 10 raised to minus 8 to 10 to 10 raised to minus 8 m reflect a high degree of specificity for the target antigen. The fact that a very large library of single-chain antibodies, all with high affinity for the target antigens, could easily be generated by this procedure means that the goal of being able to select virtually any antibody from a non-immunized library has been achieved. Chemically linked monoclonal antibodies are drugs that are very effective when tested in cell culture, are often much less potent in a whole organism. The difference in potency is typically due to this is figure 10.39, construction of large library of single-chain antibodies, B cells, from several non-immunized individuals were collected and pooled. The mRNA was isolated and used to program the synthesis of cDNA. Take a screenshot of these. Difference in potency is due to the drug not being able to reach the targeted site in the whole animal at its concentration sufficient to be effective. Increasing the dose of a drug is not the answer to this problem because high drug concentrations often have deleterious side effects. A number of different strategies are used to enhance the delivery of a drug to its target site. Excuse me. 1. Drugs may be encapsulated in liposomes, that is, particles in which the drug is surrounded by a specific lipid surface that can be targeted to certain organs. 2. Certain toxin genes may be incorporated into tumor-infiltrating lymphocytes. These cells can deliver the incorporated toxin. Directly to the site of a tumor. 3. A drug can be coupled to the monoclonal antibody that is specific for proteins found only on the surface of certain cells. So these are all different strategies used to enhance the delivery of a drug to target site. Let me repeat that for you. Drugs may be encapsulated in liposomes, that is, particles in which drug is surrounded by a specific lipid surface that can be targeted to certain organs. 2. Certain toxin genes may be incorporated into tumor-infiltrating lymphocytes. These cells can deliver the incorporated toxin directly to the site of a tumor. 3. A drug can be coupled to a monoclonal antibody that is specific for proteins found only on the surfaces of certain cells, example tumor cells. And 4. A prodrug is an inert form of a drug that requires a specific enzyme to be activated to ensure that the drug is released only in the vicinity of the target cells. The activating enzyme is coupled to a monoclonal antibody directed against specific cell surface antigens. For this type of therapeutic system to be effective, the monoclonal antibody or single-chain antibody that is complexed, but the prodrug converting enzyme must be available in quantity and relatively pure form, bind to a protein that is highly specific to the target cell, be stable under physiological conditions, but clear rapidly from circulation, and when necessary, be able to penetrate into tumor masses that all of the cells can be exposed to the drug. With this approach, only specifically targeted cells are exposed to the drug. permitting the use of a much lower concentration than if it were administered directly. Dual variable domain antibodies. In some instances, an antibody, particularly one that is conjugated to a toxin or a radiochemical, is able to destroy a tumor or pathogen cell. In such cases, it is often advantageous to use antibody fragments, since the Fc portion of the molecule is not only needed, it may impede or prevent the rest of the molecule from binding to relatively inaccessible antigens. Despite the usefulness of antibody fragments in a variety of applications, is a major limitation of using them as therapeutic agents is that since they lack the Fc portion of the molecule, they are unable to mount a complete immune response. To increase the utility of naturally existing antibodies, as well as to ensure that they are effective initiators of a complete immune response, researchers have created what they have termed dual variable domain immunoglobulins. These constructs are essential IgG molecules containing two tandem Fv regions, each with a different specificity. Dual variable domain immunoglobulins are bispecific and tetravalent, and they consist entirely of human immunoglobulin sequences. That is, each molecule contains 4 Fv regions, 2 identical Fv regions directed against one antigen and 2 Fv regions directed against another antigen. A dual variable domain immunoglobulin specific for both interleukin 12 and interleukin 18 produced in CHO cells showed binding to both of these cytokines, binding each with an affinity similar to that of the original monospecific antibody. In addition, in a biological assay, using a severe combined immunodeficient mouse model when grafted with human peripheral blood mononuclear cells, the interleukin 12 by interleukin 18 antibody was as effective at inhibiting induced IF and gamma production as was a combination of the original 2 monospecific antibodies. Using this strategy, it should be possible to generate full-size bispecific antibodies for a variety of therapeutic purposes. Then we are on to anti-cancer antibodies. A number of therapeutic antibodies that are directed against protein antigenic determinants of the surface of cancer cells have been selected because the proteins are overexpressed compared to those in non-cancerous cells. Unfortunately, antibodies directed against these proteins may also bind to some non-cancerous cells expressing the same or a similar antigen, and 2, this approach presents researchers with only a limited number of targets for therapeutic antibodies. One way to select for additional cell surface targets would be to identify proteins whose expression is selectively induced in tumor cells exposed to chemotherapeutic drugs. When colorectal cancer cells were treated with the drug irinotecan, which is a topoisomerase inhibitor and is commonly used to treat this type of cancer, several newly synthesized proteins were found on the surfaces of those cells. Topoisomerases are enzymes that unwind DNA during either DNA replication or mRNA transcription. The new cell surface proteins were expressed early, prior to any major effects of the chemotherapeutic compound on cell viability. Monoclonal antibodies directed against one newly synthesized cell surface protein called LY60E48 were generated, and then the antibodies were complexed with the cellular toxin auristatin E. The antibody toxin conjugate was then used to treat tumor cells that were first treated with irinotecan. Following binding to the cell surface, protein with antibody toxin conjugate was internalized inside the tumor cell with the strategy. In 6 out of 8 mice, tumors disappeared entirely with this strategy, while in the other 2 mice, tumors were dramatically decreased in size. With this exciting approach, will have to be tried with larger numbers of animals before it can be in as clinical trials. However, provided that it is possible to identify one or more proteins that are specifically induced by chemotherapeutic agents and are not found on the surfaces of non-tumor cells, this procedure could become a general strategy that is used to treat a variety of different types of human cancer. There's a milestone note we haven't completed. Let me just finish that for you. This is about the construction of a retrovirus packaging mutant, and it's used to produce helper-free defective retrovirus courtesy R-Man, R.C. Mulligan, and T. Baltimore from 1983. Human gene therapy has always been a controversial subject. However, most scientists have agreed with T. Friedman and R. Roblin, who in 1972, in the magazine Science stated, "In our view, gene therapy may ameliorate some human genetic diseases in the future. For this reason, we believe that research directed at the development of techniques for gene therapy should continue. The essential features of human gene therapy are the delivery of a remedial gene and its expression in a cell type or tissue that cures a disease without risk to either those administering the therapeutic agent or those receiving it. Initially, vectors derived from human viruses seemed likely to be the principal mode for delivering remedial genes because they have specialized mechanisms for entering specific cells. In particular, of the various potential viral vectors, those based on retrovirus were considered the most promising. However, a native retrovirus is an infection agent that can cause cell damage and in some instances induce cancer. The most significant advance that made human gene therapy possible was the development of a system for packaging a remedial gene into a non-infectious virus particle that retains the capability of attaching to its host cell. Mann et al. Constructed the first retroviral packaging cell line. In a sense, they integrated a viral genome from which they had removed a DNA segment that contained the packaging signal into a chromosome of a cell line. Under these conditions, the cell line produced non-infectious virus particles. However, after transfection of these cells with a DNA construct that had a packaging signal and a remedial gene but no retroviral genes, the construct was packaged into virus particles that in turn could be used to deliver a remedial gene to a particular cell type. This clever strategy was immediately adopted by many other researchers who were working on human gene therapy. Over the years, the original retrovirus cell packaging line has been enhanced and the concept has been successfully applied to other viral vectors. In 1985, as a result of the work of Mann et al., the others, W. Vanderson, who has been a persistent advocate for human gene therapy, noted, "It now appears that effective delivery expression systems have become available that will allow reasonable attempts at human gene therapy." On 22nd May 1989, Anderson and his colleagues initiated the first clinical trial using a gene therapy strategy. To date, so this is a couple of years back, gene therapy in general has not been particularly successful. However, as more information accumulates from ongoing studies, it is inevitable that it will become the standard mode of treatment for many diseases. Right, we're on to the summary of this chapter, or this topic of molecular diagnostics. A large number of in protein therapeutics. We have a large number of proteins that have potential as therapeutic agents have been synthesized from cloned genes in bacteria because most of these proteins are from eukaryotic organisms. The strategy for the isolation of a target gene often involves isolating mRNA enriched in the messenger of interest, synthesizing a cDNA library, and subcloning the selected target cDNA into an appropriate expression vector. In some instances, novel and useful variants of these proteins can be constructed either by shuffling functional domains of related genes or by directed replacement of functional domains of the cloned gene. In addition, long-acting and stable variants of some therapeutic proteins have been synthesized. In some instances, genetically engineered enzymes may be used as therapeutic agents. For example, both recombinant DNase 1 and alginate lyase have been used in an aerosol form to decrease the viscosity of the mucus found in the lungs of patients with cystic fibrosis. In addition, phenylalanine amylase lyase may help patients with phenylketonuria as a replacement for phenylalanine hydroxylase. Alpha-1 antitrypsin may be used to limit some infections, and glycosidases may be utilized to convert blood groups A, B, and AB to type O. The development of recombinant DNA and monoclonal antibody technologies combined with the understanding of the molecular structure and function of immunoglobulin molecules has provided specific antibodies as therapeutic agents to treat various diseases. Antibody genes can be readily manipulated because the various functions of an antibody molecule are confined to discrete domains. Drugs, prodrugs, or enzymes can be coupled to monoclonal antibodies or Fv fragments that are specific for proteins found only on the surfaces of certain cells, example tumor cells. These antibody drug or anti-drug enzyme combinations act as therapeutic agents. However, if the therapy requires multiple treatments, the antibody component should be from a human source to prevent immunological cross-reactivity and sensitization of the patient. To achieve this, rodent monoclonal antibodies are humanized by substituting into human antibodies only the CDRs or the rodent monoclonal antibodies. In addition, it has become possible to produce and select human monoclonal antibodies in E. coli and in transgenic mice. And we're going in for the review questions. Right. 1. Before the sequencing of human genome, how would you have cloned and expressed a cDNA sequence encoding human interferon? You do not have a DNA hybridization probe for human interferon, although you have isolated a human cell line that can be induced to synthesize IF in approximately 100-fold over background levels. Explain your strategy. Second question, what is the Fc portion of an antibody molecule, the Fab portion, Fa portion, and the CDR portion? How antibody light and heavy chains coordinatedly synthesized in E. coli? How would you modify growth hormone to make it long-acting? Why would DNase 1 and alginate lyase be useful for treating cystic fibrosis? How is the production of alginate lyase from a cloned gene defective in E. coli transformants? What is a combinatorial DNA library? How is bacteriophage M1 used to select Fv fragments that bind to specific target antigens? What are disulfide stabilized Sc Fv molecules? How are enzymes coupled to monoclonal antibodies or Fv fragments used as therapeutic agents? How are mouse monoclonal antibodies selected? Humanized. Describe the reasons for creating humanized monoclonal antibodies. Describe a protocol for producing a therapeutic agent that targets and kills a specific cell type. How would you engineer TNF-alpha to be a more specific and effective anti-cancer agent? What would you do to make interleukin-10 more effective for treating inflammatory bowel disease? How can the KORAH gene for DNase1 be manipulated so that the enzyme becomes more effective for treating cystic fibrosis patients? How would you develop a strategy to protect at-risk women from HIV infection? How might low levels of phenylalanine be attained other than the phenylalanine-free diet in patients with the human genetic disease phenylketonuria? What types of genetic manipulations can be used to generate a very large bacterial library of highly specific single-chain human monoclonal antibodies? How would you engineer a mouse so that it produced only human antibodies? I think you remember xenomouse, right? What is this bispecific diabody? How would you design a short peptide so that it retains the antigen binding specificity of an anti-immunoglobulin molecule? How would you select antibodies against specific antigens in E. coli? What are dual variable domain immunoglobulins? How can you use a chemotherapy agent to facilitate the targeting of tumor cells with monoclonal antibodies? So that's it about chapter 10 with the protein therapeutics. We'll be on the next chapter soon, and that will be titled Nucleic Acids as Therapeutic Agents, which is a subtopic of molecular diagnostics part 2. Right, so I'll see you in the next lecture shortly, and in the meanwhile you could always make notes, do some research. If you have any doubts, please get back to me on the comment section of YouTube, or to my WhatsApp, or to my email. I'm there to clarify your doubts. But I also request that you please do not commercialize any aspect of this lecture. Please use it to the best of your ability to write your exams and come out with flying colors, right? Thank you. Good day.